All right, we're getting ready for our last project. And of course, what we're doing is we're analyzing a eukaryotic gene. But um, I wanted to make sure you understood how you can find eukaryotic genes. And the easiest way, of course, is through CPG islands. Now, I explained this in class, some of you missed, but I'm going to go through this and I'm going to kind of do this part for you. And I'm going to assign you these genes and I'm going to show you how I found them. And this is something you need to include. You just don't have to do it necessarily or do it from scratch. You'll know where to look for everything. But here we have a config here. This is the FASTA file for Lonesome George. All right, Lonesome George, of course, was a turtle, a giant sea turtle, or he was actually a land turtle, but one of those giant turtles that he was the last of his kind, and for 50 years he lived on an island, and they took care of him, knowing that he was the last of his kind. And when he died, they sequenced his genome. And when you sequence a genome, the machines that do the sequencing, they give you back the FASTA files, I guess you would call it. In this, it's really not a FASTA. They convert it to FASTA, but you end up with something called a contig, and a contig is very large sequence here, and there are several genes on it, and you'd look through that to figure out where the genes are. So that's what we're going to do, and I'm going to kind of go through this for you real quick and show you how I ended up with these 4 genes. So what I'm going to do is I'm going to there are 8 of you, so I'm going to have you work in groups of 2, and each of you are going to get one of these to work on. So I'll email this out, and you'll just know that you have one of these genes, and I'll assign them to 2 of you, and you can work together. All right, so the first thing to work on is how did I find those 4? So I'm going to go to this program. This program is very similar to the DPath program that I showed you in class. It's a little better, but you could use DPath in the very much the same way that I'm going to do here. So I am going to run this, except for DPath is only going to have one color of letter, and I'm going to have multiple. So I'm going to open my program here. And I'm going to go to Lonesome George here and open up the FASTA file. And what it does is it gives me a nice graphical view. But I have a search here, down here at the bottom. And the first thing I'm going to do is I'm going to search for, in blue, C followed by G. Because what we're looking for are CPG islands, and CG will find those CPG islands. So I search for that, and they all appear. All right, back here behind me. But I'm also going to search for something else. I'm going to search for 2 CGs next to each other. Very rare sequence. You don't see this. CGs are rare enough. CG followed by another CG is extremely rare. And I'm going to search for that one on top of it. And so I'm going to close the frame. And we should be able to find our CPG islands here. Now I scroll down and, oh, there's one right here. Okay, scroll down. Definitely one, very large one here. Let's scroll up, this thing flickers too much. All right, but very large one. I would have suspected maybe this is 2 genes, but they're just too close together. It's just got a very large regulatory region here. So we scroll down, and here's another one. See, this is very easy. These are the last 2. Now I'm going to do the last one as an example so you guys can follow along in the videos and do the same things you do for your genes. So this one won't count. Somebody's going to get this last one, which will be gene 4 over here. It's going to be a lot shorter, but really it doesn't matter because most of what you're going to be doing is in the regulatory region, which I'm going to count as 1,000 base pair ahead of the first CG. So I'm going to count here, and I go backwards 1,000 somewhere in here, and I'm going to take all of that down to the next gene, and that's how I took out the piece that you're going to get. So for gene 4, I started here, and I went backwards 1,000. All right, so down here, if you notice down here, I have a number. So wherever I'm blinking, wherever the cursor is, that's where I'm at. I'm at 66, almost 67,000, so I need to be at about 66,000. So I go back up here, and I'm at 58, so that's actually, I'm pretty sure that's not right there. 59, no, 66, so about 69, so that should be at 65,000, somewhere in there. Oh, you got to go back down here to get it refreshed. This thing is, so 58, that's 66. So we need it at 65, that's 66. 66. No, it's not refreshing. Don't know why. Okay, this says it's at 59, so we need it 58. Oh, I see. It is refreshing. It's doing exactly what it should do. All right, so you would grab it right about here. That's at 58 too. So yeah, anywhere in here, grab that, and I'm going to go down to the next gene. And I'm just going to copy this piece, and I take this piece here, and I put it into the FASTA file for you guys. Oops, wrong program. The FASTA file. Let me get it back up here. Right here. And so that would have been the last one. Yes, I started at 58 and I went forward and to the next gene. So that's gene 4. I did the same thing for 3. It started at 45, roughly 45,000 and moved forward, and then 28. So these are different sizes because I go all the way to the point where the next gene starts. Gene 4 was pretty short, and now realize that the gene probably only goes somewhere in here. I see a bunch of T's here. These are probably polyadenylation signals or something here. So the gene probably only goes to about here. Most of what you're going to be doing, though, you will be finding that most of what you're going to be doing are going to be in these like 3 or 4 lines here. I have now realize these are longer than the ones you'll see in your whatever you're doing. You'll probably have to go and chop it up a little. But the 1,000 base pair between here and up here, this is where most of your regulatory, your promoter, pre-promoter region, that's where the things you're looking for are probably at. Your transcription start site will be somewhere in here, and your AUG, your start codon, will be somewhere in here, and your stop codon will be somewhere down in here, and your polyadenylation and termination signals will be down in here. So this is a smaller one. Now some of you are going to get much larger ones because I go to the next CPG island when I choose a gene, and there are probably genes in between there. So here, we're going to start somewhere up here in this one. Here's your CPG island, which is huge compared to the others, and which is fine. And then it just keeps on going until it's quite a bit of distance there. Realize there are probably some non-CPG island genes in between there. So that's going to make it rough on you knowing where to stop. So the people down here at this last one, you kind of got an easy one because you're pretty sure there's only one gene in here. The beginning, you're going to find your C's and G's, of course, which I just did for you. And then, at the end, you're going to find all your termination and polyadenylation signals, and I see them right in here. So for this one, it's pretty easy. The next one, you don't know where to stop. Now, it can't be too far down, but, a couple of thousand, 5,000 at the most, I would say, down past here. So, it's not going to go much further than, right in here. But you'll have to find it, I suspect. Let's see if I can see some T's. That looks like a termination signal right there. Could be here. Could be there. But the whole idea is you need to find that for whoever has gene 3. Now gene 2 up here, another big one here. Or no, that's the one we were in. This is even longer. Doesn't have as strict or as many CGs, but definitely it's there. So you go from here down, and again, you'll have to figure out where to stop. Probably somewhere in here. It's going to go like way down, but you don't have to go that far. You just need to figure out where your first stop is. So that's really the hard part here, is you're going to have a little bit more sequence than you need, and there are some other genes in there. You got to pick where the first one ends. Now, I didn't do the top one because there are ends. Realize the sequencers are not perfect, and there were a lot of ends up here. It looks like there's probably a gene in here, and this is a regulatory region. We can't use this piece, or we can't use this gene because this is the part that you're interested in right here, and it's not available. So whenever the sequencer can't sequence something, it just puts N for unknown or not available, whatever. Now I'm doing, as an example for you guys, I'm going to do the last gene. So we come down to the last gene. Let's scroll up a little. And it also has some N's, but it's in the middle of it. So I'm doing the last one. Well, let's get down there, wherever it is. That's not it. It's the one right after that short one. Here. So I'm doing this one for you. So I'll be giving you examples based on this last one here. And you'll get lots of videos. So I'll be sequencing this one. You guys will have this one and then the three above it. And there'll be two of you working on each sequence, and you can work together. So that'll be your last project, and that'll be due, of course, like the last day of class or whatever. So just getting you started on this. If you don't have the handout, I'm going to need to email it to me. We're probably only going to meet one more time, like after Thanksgiving. So I'll giving you more information on turning in the LAC Operon assignment. That's really the hardest one because you have to read the articles and figure it out. This one is all about searching for things and finding them in here, which you do a lot of that in the Operon as well. So you do your best on the Operon. That one, on the Operon one, you look through the articles and you can probably find things. And you can figure out where things are, even though that's a hard one because you got to read the articles and then you got to figure out what's going on, and I understand all of that. So that's where the hard part is there. But most of the articles are going to tell you kind of where things are. The thing about this one is nobody's ever looked at these sequences. So these are brand new. But I'm going to go through and I'm going to show you how to get some clues by looking at organisms that are similar to this one. And by getting clues from related organisms, we can figure out exactly where things are so you can test your analysis. Now this is pretty easy. You go through the little handouts I gave you, which is more than enough information to find things here. And you can find the things, and then you can go compare it to another organism, the same gene, and you should be able to kind of check yourself, because we can use what's called the green sea turtle, which is a related organism, and we can compare the genes here with the genes there, and you should be able to like figure out that, oh, yep, I got it right, or I didn't. So I'll talk more about those things later, but I just wanted to show you how I picked out your genes that you're going to get for your final assignment. And if you weren't there the last time, you need to find out about DPath from me, and you need the handout that tells you how to go find all these pieces. Should be pretty straightforward with the handout, but we'll see.